Welcome to the screencast on vaccination, and also there's a little bit about discovery of drugs. Okay, so the first slide here looks at comparing different types of immunity. Okay, immunity is ability to be able to avoid a particular infection or avoid the symptoms of a particular infection, and so there's various types. There's artificial and natural, both active and passive. Okay, so the active bit here basically means that you have at some point been infected with antigens, okay, either naturally or artificially. Okay, naturally is what we've looked at in previous screencasts, which is when you've actually had an infection from the actual pathogen with particular antigens on it, and you have as a result created memory cells to that, and therefore have become immune to subsequent infections of that disease. Okay, so the chickenpox example that I gave you, the first time you get chickenpox, you get one of those primary responses, create those memory cells, and then you're immune to chickenpox thereafter. Okay, because you've got those memory cells which have built up in your circulatory system and your lymphatic system, which will be able to recognize any subsequent infections of chickenpox and therefore make you immune to it. Okay. However, there we've got active immunity, which can be artificial, which is when we deliberately inject you with some attenuated or dead pathogen, okay, which has got antigens on it, which you can't actually get the disease from, and what happens is that you create a primary immune response to that as well, and create memory cells so that if you ever do encounter live versions of that pathogen, you'll be immune to it. Okay. There are then passive immunity, and passive immunity, which is when you receive antibodies, okay, from various sources. Okay, so naturally passive or passive natural immunity is happens in pregnancy, when antibodies will pass from mother to developing fetus across the placental wall. Okay, they also can occur in breast milk. Okay, so the newly born baby can get some passive natural immunity if he or she is breastfed. Okay, i.e. Diseases which the mother is immune to, the developing child will also be immune to. Okay, or there's passive artificial, which is when we inject you with some sort of serum for some sort of which has got antibodies in it. For example, anti-venom for snakes. This serum that we inject into people that would have antibodies in it, and that would be passive artificial. Okay, so these are various types of immunity. You've got to be able to understand the differences between these, and here I've got the whole thing just represented diagrammatically. So naturally, okay, active is antigens after you've actually had a disease. Okay, so you've had a primary response and got memory cells. Okay, so that's what happens naturally. And then passive is when a developing fetus receives antibodies through the placenta, or a young suckling child gains antibodies from the mother's milk. Okay, whereas artificially, okay, that can be active via vaccines or serum which has got antibodies which we deliberately inject into a patient for some reason or another. Okay, so those are all the differences between naturally active, naturally passive, artificially active, and artificially passive. Okay, so you need to know the differences between all of those. So moving to the next slide. This slide looks at vaccination. Okay, first of all we'll look at actually how vaccination works, and then we'll look at how we can use vaccinations within populations to control disease. Okay, so it's dealing off with the vaccination. What a vaccination is, it's a whole live microorganisms, or it's something which has got antigens on it. Okay, so the famous story of Jenner deliberately injecting patients with cowpox, which the patients then become immune to. Okay, and in so doing also immune to a much deadlier disease, smallpox. Okay, so the idea is that the cowpox, by the infection of cowpox, we get memory cells, and those memory cells not only recognize future infections of cowpox, but also are able to recognize the smallpox and are able to confer immunity on the patient. Okay, or it can be a harmless version of the pathogen. So what we sometimes call an attenuated virus, for example, is a virus which has its DNA removed, so it can't reproduce and therefore can't cause disease, but it still has the copies of the antigen on its coating, and what then happens is that we create memory cells to that particular pathogen, and that can confer immunity on the patient as well. Okay. And also we can just develop antigens from the pathogen and inject those too. Okay, so those are ways that we can what vaccines are, and the idea is that we inject those into the patient, and the patient undergoes a primary response and creates memory cells. Okay, and therefore those memory cells are sitting there waiting for the individual to be infected with the live disease, and in so doing the person will be immune to it. Okay, so here we've got our antigens which we inject into our individual, okay, and with those antigens then undergo an immune response, okay, with with T and B lymphocytes, and the B plasma cells eventually produce antibodies to that antigen. Okay, so that's what vaccination is. Here I've got a animation of that.
Different ways, such as through an open wound, by inhalation, or through contact with mucous membranes of the eyes, nose, or mouth. If your body becomes infected by a pathogen, the white blood cells circulating in the blood go through a trial and error period to determine the best way to kill the organisms. During this period, the body is sick with symptoms of the illness. When the white blood cells determine how to destroy it, they create memory cells to handle future attacks of that same pathogen. Vaccination is a way to trigger the body's immune response without becoming sick. The vaccine contains weak or dead organisms that usually do not cause illness. The body is able to destroy the pathogen in the vaccine and produce memory cells. When the body develops immunity to a specific pathogen, immunization has occurred. If the body is ever exposed to the same pathogen in the future, the memory cells react quickly without a trial and error period. This allows the body to destroy the pathogen efficiently before it can cause illness.
Okay, so that looks at that's hopefully illustrated what a vaccination is. Now, looking at how we can use vaccinations to try and control disease within a population, there are two strategies that we can use here. One is called herd vaccination, which is when we try to provide immunity to the whole population, and that requires about 85 to 95% of the individuals within the population to be vaccinated. Okay, and if I look at a diagram below here, okay, so here I've got the not immunised but healthy blue individuals, immunised and healthy are the yellow ones, and the not immunised, sick and contagious ones. Okay, so if I've got a population of people who are not immunised, and we put some people who've got the disease amongst it, what will happen is the disease will spread quite rapidly and affect the majority of the people in that population. Okay, if you've only got some people immunized, these yellow ones here, what happens is that the same happens, that very rapidly we will find that the majority of the population will be able to get the disease, obviously apart from those ones that happen to have been immunized. Okay, now if we have the majority of the people immunized, as we have down here, okay, so all these yellow ones here are the people that we have immunized, okay, and what we see is that even if there is the odd person that is not immunized, okay, what happens is that because the majority of people are immunized, the majority of the ones that weren't immunized, the few that weren't immunized in here, won't get the disease, okay, because the likelihood of them encountering the disease from the few people that have the disease in the first place is far less, okay. So that's why we look to try and immunize 85 to 90% of the population, and that will confer immunity on the majority of the population, okay. So that's what we call herd vaccination. Going back here, ring vaccination, on the other hand, is when we have an outbreak of an infection in a particular area. This is particularly useful for farm animals. If we have an outbreak in a particular farm of a particular disease, what we do is that we vaccinate all of the animals on the surrounding farms. Okay, and in do so, what we do is we create this ring of vaccinated animals around the diseased animals, and because the vaccinated animals don't get the disease won't spread beyond that ring. Okay, not necessarily so useful in human infection, because obviously humans have the freedom of movement and can move out of the areas and infect other people, but farm animals obviously are contained within the particular farms that they're in, and therefore by vaccinating the surrounding farms you prevent the disease from going any further. Okay, and that would be known as ring vaccination. Okay, so next slide. This slide deals with the threat of new strains of particular diseases, and particularly influenza. One of the things about viruses is that they are constantly forming new strains. Okay, mutations occur as the viral DNA gets copied in cells, and because those that DNA encodes for antigens, the antigens change, and if the antigens change, the memory cells for previous antigens will not recognize the new antigens which have been the result of these mutations. Okay, consequently, the new strain of the virus will cause disease. Okay, so as I say, influenza caused by a virus, proteins of virus act as antigens. Antigens of the virus change, forming new strains of virus. Memory cells for one strain will not recognize a new strain. Okay, so with influenza, every year different strains in the population thus require different vaccinations. Okay, which is and this is quite difficult to manage. So government health organisations plus the World Health Organization, what they do is they sample different strains in populations, and then the pharmaceutical industry develop new vaccines that are effective against new strains, which is a big industry in itself. And then what the governments tend to do is they implement vaccine programs to protect the populations most at risk. Okay, so those most at risk will be the very young, the very elderly, or people who are very ill, plus primary healthcare workers, doctors, nurses, who are constantly in touch with people that will be exposed to illness. Okay, so that's what happens with respect to how we try to deal with new strains of influenza each year. Okay, moving on to the next slide. This last slide looks at new sources of medicines with reference to microorganisms and plants. Okay, many new drugs are constantly required for new and untreatable disease. Okay, as well as the idea of antibiotic-resistant bacteria, which we've come across before. And now a lot of drugs are actually isolated from living organisms. So the great story about Fleming and his penicillin, found from his fungi growing on his plates back in the late 20s. Okay, is an example of where a naturally living organism has produced a phenomenally valuable drug. Okay, other drugs that we've talked about in previous slides, like the streptomycin drug, which was used to be the most effective antibiotic against TB, again was isolated from living microorganisms. Okay, other drugs can be purified from traditional herbal medicines. Okay, aspirin was an example of that. Other drugs that have come from plants, the drug that we talked about in terms of the one that can control the malarial parasite, the one that renders hemoglobin toxic to malarial parasites, a drug called quinine was first isolated from the bark of a tree called the cinchona tree, which was found in South American rainforests. Okay, so these are all examples whereby very valuable drugs have been isolated from living organisms, be they microorganisms or plants. Okay, and that links in with another area of one of the, on this f212 module, the importance of maintaining species biodiversity. Obviously, if we didn't have this species biodiversity, we wouldn't have the possibility of isolating new molecules which are present in the environment in the future to be able to cope with either antibiotic-resistant bacteria or other untreatable diseases which we might not even know about yet. Okay, I've also put in here a bit about genomics, which is a new science of trying to understand the shapes of proteins, okay, and how they interact with molecules in the body, and understanding what genes are required to encode for these particular proteins. So looking at the shapes of molecules and designing shapes of molecules which might be able to have some form of efficacy as a drug, okay, because many drugs are designed to block some sort of receptors. Okay, this whole idea of cell signaling that we've come across, if we could interrupt cell signaling or manipulate the whole cell signaling process, that is how many drugs work. And if we could actually know what the receptors look like, and therefore know what the shape of drugs that would interact with those receptors, that is the whole field of genomics. Anyway, that finishes our screencast on vaccination and possible sources, new sources for medicines.